Now we're going to continue with a different group of anaerobes, and we're going to discuss the anaerobic gram-negative bacilli. The genera that are going to be discussed in this section are Bacteroides, Prevotella, Porphyromonas, and Fusobacterium. Now, we already mentioned that Bacteroides is the most commonly isolated anaerobe in the clinical microbiology laboratory. Now, this slide shows you the breakdown in a pie chart of the specifically gram-negative anaerobes. And you can see Bacteroides fragilis is 61% of all of the gram-negative anaerobes. So not only is Bacteroides the most commonly isolated anaerobe, but Bacteroides fragilis, that specific organism, is the most commonly isolated anaerobic organism in general. The general features of this group of organisms, the gram-negative bacilli anaerobic bacteria, are that they're pale staining. They are gram-negative rods. Some of these organisms are encapsulated, and they are anaerobic to somewhat aerotolerant. So on the top left is a gram stain of Fusobacterium nucleatum, and Fusobacterium is appropriately named because it has a fusiform shape to it. So if you look at a gram stain of Fusobacterium, the organism is very, very thin, long, and has tapered ends. On the bottom right, this is a different species, Fusobacterium mortiferum, and this has various shapes to it. So Fusobacterium nucleatum is the most common Fusobacterium of the anaerobes, and it has that fusiform shape, but not all of the Fusobacterium organisms have that characteristic fusiform shape to it. Now, the clinical manifestations of the gram-negative anaerobes. Many of these organisms cause oral and dental infections. So remember, we said that our anaerobic bacteria, just like our facultative bacteria, can be normal flora. So a lot of these organisms are normal oral flora. They actually contribute to cavity formations, as does a lot of our other facultative organisms. So these organisms can live deep in the gum tissues where there's less oxygen, and can cause plaque and cavities just like a lot of the other bacteria can. So our oral and dental areas of the mouth, we have anaerobic organisms there. We have a lot of it's a polymicrobic environment. A lot of the infections that we will get in an oral or dental area are going to be endogenous. They're going to be from our own oral flora, and many times, as we've said, they're going to be polymicrobic. You're going to get an infection with multiple organisms. Many times they remain localized, so within that oral area, but they can disseminate. So the organisms that are commonly involved in oral and dental infections include Fusobacterium, the non-pigmented species of Prevotella, as well as the pigmented species of Prevotella and Porphyromonas. These types of anaerobes can also cause throat and pharynx infections. Specifically, there's something called Vincent's angina, which is an ulcerative lesion in the pharynx. Many times this is caused by an infection with Fusobacterium necrophorum mixed with treponemal organisms. So this is a very specific type of throat and pharynx infections, and it's not really a common infection, so it's not like it's something you would see all of the time. With our anaerobes, you can see bite and wound infections, and those definitely tend to be polymicrobic because your bite infections are caused by oral flora, and your oral cavity has a lot of different microbes in there, facultative organisms mixed with anaerobes. So a lot of times you'll get a polymicrobic bite infection. The same with wounds. You cut yourself, the deeper tissues, you might get an anaerobe that's able to grow in that area with your facultative organisms. So again, your facultative mixed with your anaerobes. The common anaerobes that you would find involved in bite and wound infections are Porphyromonas, Prevotella, Bacteroides, and Fusobacterium, which is pretty much all of the gram-negative anaerobic rods that cause human infections. Lung and pleural space infections, you can get aspiration pneumonia. Now, we've discussed aspiration pneumonia before with our facultative organisms. Commonly, aspiration pneumonia is caused by our Enterobacteriaceae family members, but you can occasionally get a polymicrobic or anaerobic infection. More commonly, if your anaerobes are involved, it is going to be an abscess. So this would be a lung abscess, can cause abscesses in individuals with underlying lung diseases like emphysema, and the organisms that might be involved are Prevotella, melanogenicus, and Fusobacterium nucleatum. The female genital tract does have some normal flora in it. Both our facultative organisms that we've talked about in the past, like our lactobacillus, but there are normal anaerobic flora that can cause, like our other organisms, pelvic inflammatory disease, chorioamniotic infections, endometriosis, bacterial vaginosis. If you remember, we talked about bacterial vaginosis when we talked about Gardnerella vaginalis, and how if your lactobacillus is depleted due to antibiotic ingestion, you can get overgrowth of Gardnerella vaginalis that can cause bacterial vaginosis. Well, you can also have the same thing happen with your anaerobic organisms that can be normal flora in the genital tract. So the organisms involved are Prevotella, Bacteroides fragilis, and the pigmented Porphyromonas. Intra-abdominal infections. Now, we do have a lot of anaerobes as well as our facultative organisms. Again, a lot of our Enterobacteriaceae members are normal flora of the intestines and bowels. So our anaerobes that would be involved in abdominal infections would be Bacteroides fragilis, as well as the other Bacteroides species that are the non-fragilis Bacteroides species. Any type of abscess can be caused by an anaerobe. So you can have a brain abscess caused by Prevotella and Fusobacterium. Cardiovascular infections, you can have bacteremia, endocarditis. So the organisms that are commonly involved in the cardiovascular infections with anaerobes are Bacteroides fragilis, and this is usually when your normal gut flora somehow get into the bloodstream. So this can be due to a perforation. This can be due to a ruptured hernia in the intestinal wall. I know someone who was getting a colonoscopy where you have a scope and to look at the intestines, and during the colonoscopy they perforated the individual's intestine, and they had to do emergency surgery right away. They had to stop the colonoscopy and get that person into the operating room A perforation of an intestine is a life-threatening situation. You have so many organisms in the gastrointestinal tract that if there's a perforation, those are getting into your bloodstream, and there's so many organisms, it can cause bacteremia, which can lead to septic shock, which can lead to death quite rapidly. Other organisms are Porphyromonas, Prevotella, Bacteroides, or Fusobacterium. Basically, any organism can cause a cardiovascular infection. These are the organisms that are normal flora in the gut as well as in the oral flora. So remember your oral flora. If you have a pre-existing condition, if you're immunocompromised in any way, your oral flora can get into your bloodstream and can cause endocarditis. Remember, haystack organisms, common causes of endocarditis. So at this point, I'm hoping that, I keep saying this, but I can't stress it enough. It's so easy to focus on the organisms we're talking about this week, but you really want to be making those connections to the previous lectures, the previous weeks, all of the other organisms, because hopefully by the time you end Microbiology 1, you will have a good idea of what organisms are out there in our bodies, what organisms are normal flora, what organisms are never normal flora. When you know which organisms are normal flora in the specific regions of the body, you have a better idea of what types of infections they can cause. When you get clinical specimens in the clinical micro lab, you already know which organisms you should be looking for because you already have an idea of what might be there. So, okay, back on track. Natural habitat. Now, okay, Bacteroides, we already said, normal flora of the colon as well as the female genital tract, which is why Bacteroides can commonly cause bacterial vaginosis. And again, if you get that perforation in the colon, you can get Bacteroides in the bloodstream. Now, Prevotella and Porphyromonas are normal oral flora, the human mouth. Can be flora in the urogenital tract as well as the intestinal tract. And Fusobacterium is normal oral flora, the colon, as well as the genital tract. So you can expect to find these organisms in the body. They don't commonly, Bacteroides is a more common cause of human infection. The others aren't as common, but if the opportunity arises, any of these organisms can cause infection. So transmission, we've said it several times. I'll say it again. Endogenous, usually with your anaerobes, you get infected with your own normal flora. Many times through bites, through trauma, through a perforation, things like that. Controlling, individuals are at lower risk of an anaerobic infection if they are immunocompetent. Anytime you're immunosuppressed, it's going to increase your risk, as well as preventing trauma, bite wounds, things like that. Now, treatment. This is important. This is a red star. Bacteroides fragilis is highly resistant to antibiotics. You must know that. It is always resistant to penicillin. You don't even test it. You don't even do an antibiotic susceptibility for penicillin with Bacteroides fragilis. It's a waste of time. It's commonly resistant to tetracycline. It's usually susceptible to clindamycin and metronidazole, but you do have to test it because Bacteroides fragilis can become resistant pretty quickly to various antimicrobial agents. Resistance is also becoming more commonly found in other Bacteroides species, the non-fragilis species, in your Porphyromonas, in your Prevotella, your Fusobacterium, your Clostridium, all of these anaerobes that many of which used to be susceptible to all antimicrobial agents are now starting to become resistant. The laboratory diagnosis of your anaerobes. Of course, you always want to inoculate your appropriate aerobic and anaerobic media. I can't stress that enough. You always are going to set up both an aerobic and anaerobic set of plates when you get a request to set up an anaerobic culture. You're going to do both. You always want to set up some sort of blood agar, and it's going to vary depending on the laboratory you're in. Some labs are going to use sheep blood agar, some are going to use Brucella blood agar. Thioglycolate broth. Not all labs will inoculate a thioglycolate broth, but some will. And then you will commonly use some sort of selective media. So Bacteroides fragilis is the most common anaerobe isolated from clinical specimens. So a lot of times clinical labs will use a selective media specifically for Bacteroides. Commonly, at the very least, you will use a selective plate that contains kanamycin and vancomycin, which will help you select Bacteroides. But there are specific selective media that are only made for Bacteroides, so some labs will use that. You're going to incubate your anaerobic set in an anaerobic atmosphere. You're going to incubate your aerobic set in the CO2 incubator. And your anaerobes you will incubate for at least 48 hours. You also are going to want to do your susceptibility testing. You could do your presumptive testing where you're dropping your antibiotic discs onto your plates when you inoculate. You can inoculate drop a bile disc. You can also use a bile broth to see if your organism will grow in the presence of bile. You can look for pigmentation. You can look to see if the organism pits the agar. You can do urease testing. You could see if it fluoresces under a UV lamp or light source. But to definitively diagnose, we always want to rely on our biochemical tests, the analysis of the end products or molecular techniques. You always do want to look at that gram stain morphology. You want to, this group, the gram negative rods, usually are pale, very irregularly stained. You could have your long fusiform rods. You can have your tiny rods, your Coccobacilli, that would be your pigmented Bacteroides. You could have some bizarre shapes. The Fusobacterium organisms tend to be bizarrely shaped. Now, some strains of Porphyromonas and Prevotella will fluoresce, a brick red fluorescence when they're exposed to a UV light source. And we already talked about some of the antimicrobial discs that you can drop when you're inoculating your specimens onto your blood-based media. And then you're going to look for the zone of inhibition. If there is a zone, the organism is susceptible. If there is not a zone and the organism is able to grow right up against the disc, it means that that antimicrobial agent, the organism is resistant to it. Here's a gram stain, Fusobacterium, nice gram negative, tapered ends, nice fusiform shaped organism. Here's Bacteroides fragilis, the most common anaerobe isolated in the clinical laboratory. Now, it is not the most common bacteria isolated in the clinical laboratory. It's the most common anaerobe. So the top left is the gram stain of it, kind of a short gram negative rod. On the bottom right, there is the KVLB, which is kanamycin vancomycin laked blood agar on the left-hand side of the plate, and on the right-hand side is the Bacteroides agar, which is the BBE agar. So you could see your Bacteroides species is going to grow nicely on the kanamycin, vancomycin laked blood agar because it's not inhibited by kanamycin or vancomycin, whereas some of the other anaerobes wouldn't be able to grow on that agar. And then on the BBE agar, Bacteroides fragilis turns the media a dark brown color. Again, a gram stain of Bacteroides fragilis. This is a small coccobacillus to short rod. Now, in addition to being highly resistant to antibiotics, Bacteroides fragilis is resistant to bile. There are some other Bacteroides species that are in the fragilis group that are resistant to bile. Now, other organisms, anaerobes, are sensitive to bile. So it's really important to remember that Bacteroides fragilis and the members that are considered to be within the fragilis group are highly resistant to antibiotics and resistant to 20% bile. The non-pigmented Prevotella, the pigmented Prevotella porphyromonas, the non-fragilis Bacteroides species are all sensitive to bile. We're going to move on to the last part of this lecture.